Hey, welcome to this video. This is what you can expect from this video in the first part of immune system. Let's begin from basic. Immunity is the capability of our body to protect us against harm. And immune system is the collection of cells and molecules responsible for defending the body against the pathogenic microbes in the environment. To understand the immune system, we should be familiar with few terms. Antigen, substance, usually protein in nature, which when introduced into the tissue stimulates antibody production. This could be a part of bacterial structure or certain protein in food or components of RBCs. In autoimmune diseases, when body is not able to distinguish self from non-self, body could destroy their own cells. So, these antigens are called autoantigens. Hapten is a non-protein substance which has no antigenic properties, but on combining with a protein can form a new antigen capable of forming antibodies. So, basically it cannot induce antibody production on its own, but combined with another protein complex, following which only, it can induce antibody production. One well-known hapten is urushiol, the toxin of poison ivy. When absorbed through skin from a poison ivy plant, urushiol undergoes oxidation in the skin cells to generate the actual hapten, called a quinone, which then reacts with skin proteins, induces contact dermatitis later. Another examples include hydralazine causing lupus erythematosus and penicillin class drugs causing autoimmune hemolytic anemia. Antibody, protein substance produced as a result of antigenic stimulation. Globulin, that we so commonly hear of, as circulating antibodies. Let's have a look at what our immune system should be doing. First and very important function of immune system is to differentiate which is the self antigen and which is foreign or non-self, and then remember it, on next exposure, to save time. And the most important function is, of course, to kill these harmful substance, either by antibody production or by cell-mediated process. A problem in differentiating self from non-self can result in autoimmune disease, in which the immunity destructs its own cell because it labels them as foreign particle. A problem in cell-mediated immunity or antibody formation can lead to immunodeficiency. If immune system acts more vigorously than it should, we encounter hypersensitive reaction. We have two types of immunity. One we are born with, that is innate or natural immunity. Second, that we develop ourselves, which is called acquired or adaptive immunity. Natural or innate immunity depends upon the genetic constituent and physiology of a person. Acquired immunity develops following an exposure, which can be due to infection or from vaccination. Natural, innate immunity is present since birth. And acquired, as the name suggests, is acquired later in life. Since this is innate immunity is not formed against any particular microbe, it is non-specific. And acquired is specific as we develop it following exposure to a particular antigen. Natural immunity acts as a first line of defense and acquired usually comes into action second. Since natural immunity is non-specific, it is less potent and acquired is more potent. In natural immunity there is no memory of previous exposure, which is present in acquired immunity. Natural immunity is faster to act and acquired is slower, in comparison. Examples of natural immunity includes epithelial barriers of skin, gut and respiratory tract. And acquired immunity involves cells like lymphocytes. Both natural and acquired immunity can be further divided into humoral or cellular type. Complement system is a type of humoral natural immunity. Cellular natural immunity includes cells like neutrophils, macrophages and natural killer cells. Acquired humoral immunity refers to immunity by various antibodies. Acquired cellular immunity involves cell-mediated effects by T cells. Now coming to organs of immune system. Primary and secondary lymphoid organs. Primary lymphoid system is where immune cells are produced and mature. In secondary lymphoid organs, further differentiation and activation of these cells occur. There are two primary lymphoid cells: bone marrow and thymus. Secondary lymphoid organs are lymph nodes, spleen and mucosa-associated lymphoid tissue. The important cells of immune system are lymphocytes, monocytes, macrophages, mast cells or basophils, neutrophils, and eosinophils. Neutrophil is the major cell of immune system, constituting around 40 to 75%. Lymphocytes are the master, or the most important of all the cells in the immune system. Develop from common lymphoid progenitor cells in the primary lymphoid organs. Lymphocytes are mainly T or B type. T cells constitute the majority of lymphocytes, mainly 75 to 80%. T cells are called so as they mature in thymus, whereas B cells mature in bone marrow. T cells are responsible for cell-mediated immunity and B cell for antibody-mediated. T cells are responsible for cell-mediated immunity and delayed type of hypersensitivity. They function by interacting with other cells to kill infected cells, or activate phagocyte or B lymphocytes. T cell markers are CD3, CD7 and CD2. In case you are not familiar with the term CD, which stand for cluster of differentiation, and is used for identification and investigation of cell surface molecules providing targets for immunophenotyping of cells. These are used to identify them. T cells have receptors TCR for recognition of MHC molecules. Peptide antigens presented by MHC molecules are recognized by the T cell receptor, which activates the T cells to act further. There are three subtypes of T cells. T helper cells, abbreviated as Th cells, Q similarly, T suppressor cell, abbreviated as T-S cells. And last type is natural killer or NK cells. T helper cells secrete cytokines that help B cells to produce antibodies, and macrophage to destroy phagocytosed microbes. Thus, they help to regulate the function of other types of cells, which is why they are also called T regulatory cells. T suppressor cells suppress immune reaction, also known as cytotoxic T cells, as they destroy virus-infected cells, tumor cells, etc. Natural killer cells are large granular lymphocytes. They recognize antibody-coated target cells and kill antibody-dependent cell-mediated cytotoxicity. T helper cells are mainly effective against viruses, tissue transplant reaction, tumor lysis. Cytotoxic T cells kill virus, foreign and tumor cells. Natural killer cells are highly effective against virus and tumor cells. T helper cells are also called as CD4 plus T cells, depending upon the CD cluster on it, apart from the usual T cell markers. T suppressor cell are also known as CD8 plus T cells. Natural killer cells have CD2, CD16 and CD56 on its surface and differ from other T cells in not having the usual marker, CD3. T helper cells or T-H are further divided into T-H1 and T-H2. T-H1 secrete interleukin-2 and interferon gamma. T-H2 secrete interleukin 4, 5, 6 and 10. Let's look at B lymphocytes now. B cells are responsible for humoral or antibody-mediated immunity. B cell markers are CD19, 20, 21 and 23. Have B cell receptor for immunoglobulin IgM and IgG and also have Fc receptor for antibody attachment. T helper cell help B cell by secreting interleukins. Coming to monocytes and macrophages. After monocytes are formed and released into blood circulation, it stays in circulation for 3 days. Once they are taken up by different tissues, they are termed macrophages. In different tissue, the name of macrophages differ but function is similar. For example, in skin it's called dendritic cells. In lungs have alveolar macrophages. Reticulum cells in bone marrow. Kupffer cell in liver. Gingival macrophages in lymph nodes. Epithelioid cells in granuloma. Microglia in brain. Macrophage help in recognition of antigens and differentiation of self and non-self. They secrete cytokines, prostaglandins and acute phase reactants. They engulf microbes, which is called phagocytosis, and act as antigen-presenting cells. Basophil and mast cells are interchangeable terms used. Basophil term is used when they are in blood circulation and their counterpart in tissues are called mast cells. When immunoglobulin E, bound to antigen, bind to eosinophil surface, it causes release of granules from mast cell. Mast cell granules contain active substances like histamine, chemical mediators like prostaglandins, leukotrienes, etc. These substances released from mast cell are responsible for inflammation in allergic reactions and wound healing. Neutrophil constitutes majority of immune cells. They contain three types of lysosomal granules, on the basis of their affinity for dye. Primary granules or azurophilic granules contain hydrolytic enzymes and myeloperoxidase. Secondary granules have lactoferrin and tertiary or gelatinase granules have matrix metalloproteinases. These products help in mediation of inflammation or degradation of phagocytosed materials. Their function is almost similar to macrophages but being smaller, they are called macrophages. They engulf the microbe by the process called phagocytosis. The ingested material fuses with lysosome to form phagolysosome. Lysosomal granules then release various enzymes to kill and degrade it completely. Their function is limited by the type and size of microbe they can kill. For example, they can kill bacteria and other small materials, but not viruses or other larger particles. Eosinophils are immune cells mainly effective in parasitic infection, allergy or cancer. They contain granules containing substances, which are released when an IgA comes tagged with the corresponding antigen. These granules release the active product which help body to react against them. Cytokines are immunomodulating substances secreted in response to various stimuli. The term cyto means cell and mines means movement. Cytokines are cell signaling molecules that aid cell-to-cell communication in immune responses and stimulate the movement of cells towards sites of inflammation, infection and trauma. When cytokines are released by activated lymphocytes, they are called lymphokines, and monokines, if they are secreted by monocytes. Chemokines are chemoattractants for inflammatory cells. Based on their mode of action, they are classified into autocrines, paracrines or endocrines. They are called autocrines, if they act on the same cell which has produced it. Paracrines, if they act on cells which are close to where they are produced. Endocrines, if they travel through circulation to reach target cells, far away from they are produced. There are many cytokines present in humans. But the important ones in acute inflammation are interleukins 1 and 6, TNF or tumor necrosis factor. Important are cytokines involved in chronic inflammation are interferon gamma and interleukin 12. Interleukin 17 is produced by T lymphocytes and other cells and help to recruit neutrophils to the site of inflammation. Main function of interleukin 1 and TNF-alpha is to express endothelial adhesion molecule, to help in migration of leukocytes to site of inflammation. They are also responsible for systemic features like fever. Interleukins 1 and 6 stimulate production of acute phase proteins. Interleukin 6 stimulate growth and differentiation of T and B cells. Interferon gamma, interleukin 12 and 17 all basically help other immune cells in performing their function. If you need details, it's given in the table. Antigen-presenting cells are cells that can capture an antigen and present it in conjunction with MHC molecules on their cell surface, thus, help by bringing the needed antigen to T cell. T cells recognize these complexes using the TCR T cell receptor, and further action by T cell takes place. Major antigen-presenting cells are macrophages, B cells, and dendritic cells. A short 3D animation video on how this happens is present on the channel, link below in description. Thank you for being with us. Second part of immune system will show exactly how cells help in inflammation process in